My name is Kristen Johnson, and it's nice to be here and be able to tell you about one of the projects, which is in our group at Calibr, which is a division of Scripps Research, where we've been focusing on drug discovery and identifying ways to improve the joint and the liver through regeneration. I'd like to start today by thinking about osteoarthritis, which is a disease of aging, which you may or may not heard of, but it is a huge unmet medical need. Patients with OA progress through the course of their lives, and their joints have been injured through running or various different overuse, but also just during the course of getting older. Approximately 40 million people actually suffer from osteoarthritis, and these patients do not have any therapeutic option that they can take other than some things which may or may not alleviate pain. Unfortunately, these patients often progress and have to move towards a surgical procedure, which is very invasive and can allow, through a replacement of that joint, more movement and seamless ways of life. But what we're looking to do here at Scripps Research, and what we've already done, is invented two different paradigms by which we may be able to potentially improve the lives of patients. So this started about many years ago in 2012. K34 was actually one of the first new paradigms for OA that was invented. We actually started by running a small molecule screen and identifying a molecule which we named cardigenin. This molecule was published and now has been widely followed up by many different researchers in the field. What we did here at Scripps is we actually took that drug, optimized it so that it could be ready to be injected into patients. And now we've actually completed a phase 1 clinical study, which I'll show you some of the results from in just a moment. But what we did before we moved into patients was we looked to understand a little bit about the mechanism of action. And we found that K34 is really a new paradigm because it directly targets the endogenous stem cells and leads to the differentiation of those cells into healthy articular cartilage. You see, in your joint, we have bone and cartilage and the synovium, which all plays a role in terms of leading to that injury or the pain that the patients experience. So with K34, we can not only provide appropriate differentiation of the cells, we found that it can actually activate cartilage gene transcription, it can promote protection of the existing articular cartilage, and it leads to an effective repair in both rodent and canine animal models of osteoarthritis. What we also found is that cardigenin itself can reduce inflammation and lead to osteoarthritis pain relief, at least in rodent models. So let me tell you a little bit more about the mechanism by which K34 is working. As I mentioned, it targets the endogenous progenitor cells, and these are cells which normally exist in articular cartilage. They have a role of helping to slightly repair the joint under normal wear and tear. But unfortunately, as we age, their ability to do this is limited, and it actually goes awry. In a sense, they no longer produce the right type of matrix. So what we did is we capitalized on that by using K34, because what we do is we actually inject it directly into the articular space, so into the knee joint itself. And this then basically directs the chondrocytes to become healthy cells and begin to secrete the appropriate matrix. This can be used on its own in a phase 1 clinical study, as we did, as well as it could complement other stem cell-based therapies, such as autologous or allogenic stem cells, and work in a paraquin manner, which is also another approach for osteoarthritis that's being investigated in trials. So let me give you a glimpse of what we found in our phase 1 study. We were able to complete last year in 2020 a 60 patient osteoarthritis study. And I'd like to just thank the California Institute for Regenerative Medicine. They've actually sponsored this clinical trial, as well as much of the work that took place to develop K34 and run all of the non-clinical studies that needed to be done before we could reach the patients. So what we found in this study was that, first and foremost, K34 is safe and well tolerated. We had patients who received one injection of K34 into their knee, as well as patients who received 4 injections over the course of a month. In that second group, or the multiple ascending dose patients, we followed those patients we followed them for 6 months. And there we were able to see in both groups of patients that K34 had a nice and consistent predictable plasma exposure. And this tells us that K34 was behaving exactly as had seen in the animal models we tested before. In addition, K34 did not cause any treatment emergent adverse events that were dependent upon the dose or the amount of times that K34 had been injected. And so all of this is good news and allows us to know that putting K34 into humans is safe. We also began to look at what happens to patients who are treated with K34. So with this drug, we look to see how patients felt in terms of the pain, the function, and their stiffness. This was measured using a scoring system called WOMAC, which is an index of questions that osteoarthritis patients fill out either in clinical trials or quite frequently, sometimes when they go to their physicians for treatment. So in that, as you can see on the right side, with our 400 microgram dose group, we saw a trend of significance where patients over the course of time had an improvement in their function, as well as their pain, and some of the other readouts, which I'm not showing you here today. So this is great and exciting preliminary data to help us understand that K34 could potentially be improving aspects of OA patients' lives. So we dug into this a little bit further by using a response criteria that's very similar to many different knee osteoarthritis trials and how we can categorize patients who responded or didn't respond. So if you had a 40% change from your baseline, so how you felt on the first day that you tried, you came to the clinic, we saw that looking out at day 90 on the right side, patients who were in that responder category dose dependently increased along with the increase in the amount of K34 which was given to those patients. Again, this is a very early study and gives us a glimpse of some of the effects and responses that patients are seeing. So just to summarize, what we found with K34 is that in a 60 patient OA trial in phase 1, both patients who received single and multiple ascending doses of K34, the drug was safe and well tolerated by all the patients. So we are now ready to begin a phase 2 clinical study, and that's where we'll really begin to investigate the structure and the function more deeply in terms of OA. In this study being smaller and only measuring patients out for 6 months, we can't make a judgment call whether or not there was an increase in the amount of articular cartilage, and this is exactly what we're interested in doing in phase 2. But that hasn't stopped us here. We actually have been moving forward in the musculoskeletal disease area to try to find other ways in which we can continue to help these patients. One of those is with the development of an extended release formulation for K34. As I mentioned, some of our patients got weekly doses of the molecule. And of course, this is not practical for patients thinking about a chronic disease that may span the course of 2 or 3 decades of their lives. And secondly, we've also started targeting a cathepsin K inhibitor because this will repair not only the cartilage, which is what K34 can do, but also targets improvement in the subchondral bone. So let me tell you about both of those options briefly on the next 2 slides. First, as I mentioned, K34, because of its short half-life, really needs to be injected more frequently. But patients and rheumatologists, those physicians who would be giving these IA injections, prefer if they only need to come in about 4 to 5 times a year for a therapeutic treatment. So what we've done at Scripps is we've actually already invented 3 different formulations where using PLGA and co-formulating it with our drug K34, we're able to increase the half-life and see the exposure out to at least 75 and even out to 90 days. We took one of those formulations and we've investigated those preclinically. So at the bottom of the slide on the right side, you're looking at a efficacy model in rats where a medial tibial degeneration score was improved by a reduction in the total score, as you see with the arrow, when treated with an extended release formulation. This was very, very similar to what's seen by other competitors, as well as what the drug which we directly delivered in the phase 1 study. So we're really excited to be able to move K34 forward, either through an extended release option or through continuing studying the immediate release version, which we tested in the phase 1. But that doesn't negate the fact that we know other patients may have other skeletal diseases, which could be challenging, as well as have a need for improvement in the growth of their subchondral bone. So you can see in the gray part of the cartoon that the osteoclasts and the osteoblasts help make up the subchondral bone. And this is something that also can become injured or degraded over time in patients. So one of the targets we're interested in investigating and have already started and identified inhibitors in this pathway is we're able to look and see at cathepsin K. It's an enzyme which actually breaks down the bone. And we've also found that when you knock it down, this leads to an increase in the production of matrix, both in cartilage and bone. So we're really excited, and hopefully next time I'll be able to tell you more about how we've progressed this therapeutic treatment to go in combination or as a surrogate along with K34. So let me tell you, for the last couple minutes, let's think about transitioning into the oncology realm and patients who may need a liver resection and then, of course, need their liver to regrow. There's many patients who have hepatocellular cancer or who also have colorectal cancer, where liver metastases invade much of the liver. And unfortunately for those patients, which is a high risk situation, therapeutic treatment with normal chemotherapy alone is not enough to necessarily improve the quality or the lifespan of these patients. So physicians use different types of liver resection procedures. Some of these actually involve a 2-stage procedure where, because of the time that it takes to need to regrow the liver, that we're able to, they have to go off of chemotherapy. What we're looking at to do at Scripps with our regenerative drug, which improves liver regrowth, is to actually aim to shorten the treatment for those patients. The exciting part about that is it may open up the opportunity to allow more patients to undergo this surgery. So let me tell you a little bit about this molecule that we've invented. It targets something called MST1, and this is a kinase which exists in the YAP signaling pathway, which is one that is quite well known to be able to increase organ size, also mediates cellular stress, and limits apoptosis. So it promotes survival of many different tissues, but particularly in the liver. It's been shown already that if you knock out MST1 and MST2, that liver regeneration in aged mice, which really doesn't happen on its own, can be restored. And even further, others have taken small molecules and shown, after removing a portion of the liver, that the MST1 inhibitors can drive this regeneration and growth. So what we did here at Scripps is we actually did a high throughput screen to identify MST1 inhibitors. And we have 2 candidates which are getting close to be ready for IND enabling studies. The studies in which we really pressure test and understand if they are ready to enter into the clinic. So what we found functionally is that these MST1 inhibitors are actually able to expand hepatocytes, so the cells which exist in the liver, in an ex vivo situation. So we took our compounds and we took primary hepatocytes from rats and we stimulated them and found that we were able to increase the growth and the numbers of cells along the right lineage. And furthermore, and what I would say is even more exciting, is we were able to take mice in which we did a liver resection and monitored what happened in the case when we treated with 2 of our lead candidates for 48 versus 72 hours in vivo. We can see a nice increase in size in the weight of the liver itself, as you can see on the bottom left graph, was significantly increased with treatment of the MST inhibitors, as well as there was no significant changes in terms of liver enzymes or organ weight, other organ weights, which gives us a manner in which we may be able to proceed with this approach in the clinic. So as I finish up today, I just want to remind you that Scripps research really has pioneered many different regenerative approaches. Two of them include thinking about cartilage repair. And in that realm, we have already tested and completed a successful phase 1 with K34 and are ready to move into a phase 2 study where we begin to thoroughly investigate the efficacy of that drug in OA patients. We're expanding our effort in musculoskeletal disease to look for extended release of K34, as well as improving the lives of patients through a CAT-K inhibitor. And finally, when we think about the field of oncology and patients who are needing to have regeneration in their liver, we have been moving forward and found that MST1 inhibition can actually lead to an accelerated liver regeneration. And this may actually allow for more patients to be candidates for those procedures, which could increase their survival. First of all, I'd like to just thank all of the investigators and scientists involved in these projects. And finally, I'd like to just thank you for allowing me to share this work with you.